네, 그러면 본격적으로 오후 세션 이어가도록 하겠습니다. 오후의 첫 번째 발표는 이탈리아 파르마 대학교 약학대학 프란체스카 지메티 부교수님이 HDL 및 뇌 콜레스테롤 대사, 신경 퇴행성 질환의 새로운 표적이라는 주제로 발표해 주시겠습니다. 프란체스카 지메티 부교수님을 무대 위로 모시겠습니다. 큰 박수 부탁드립니다.
Good afternoon, everybody. I have to say that it's a real honor for me to be here, and I have to really acknowledge Rydell for this kind invitation and for the organization of the excellent symposium. In my presentation today, I will share with you some data from us and from others, suggesting how HDL and HDL-mediated cholesterol lipid metabolism may be a target in neurodegenerative disorders, particularly focusing on Alzheimer's disease. As you have seen before from the very brilliant presentation of other speakers, HDL are a very complex system. They have many proteins, lipids, they have multiple functions, and concerning the HDL cholesterol levels, all the epidemiological studies agrees in saying that low HDL cholesterol levels is associated to an increased risk of several diseases, not only cardiovascular, but also many other diseases, such as infections, autoimmune diseases, diabetes, etc. And among the multiple functions of the HDL, the most studied one, as you have heard before, are the capacity to promote the reverse cholesterol transport, so the removal of excess cholesterol from peripheral tissues to the liver, as well as antioxidant and anti-inflammatory activities. Those properties may get lost in several diseases. Among those, you can see the neurodegenerative one. And basically, what it happens are specific changes in HDL composition or density or size, leading to a modified HDL phenotype and eventually ending to dysfunctional HDL. Interestingly, HDL have been also identified in the brain, the human brain, and they have been isolated from cerebrospinal fluid from human subject. They have called HDL-like particles because they are very similar to plasma HDL, but they also present peculiar characteristics. You can see the last column at the right of this table. They have a wider size range, they are mostly spherical, and they mainly contain as apolipoproteins ApoE, which is the main lipid transporter in the brain, as well as other apolipoproteins such as ApoA1, which seems to come from the peripheral district through crossing the blood-brain barrier. Those particles have a critical role in the brain. They are transporters of cholesterol among different cells. In particular, they are transporting cholesterol, newly generated cholesterol, from astrocytes to neurons. This process is essential, especially for adult neurons, since they progressively lose the capacity to synthesize their own cholesterol, so they mainly rely on cholesterol produced from other cell sources in order to maintain their physiological functions, such as, for example, synaptogenesis or repairing of damaged membrane. This process begins with the efflux of cholesterol out from astrocytes together with ApoE to generate discoidal nascent HDL, and this occurs mainly through the activity of a specific transporter that Anand just mentioned before, which is also expressed in the brain, which is the protein ABCA1. Then nascent particles may acquire further cholesterol through the activity of another protein membrane transporter, ABCG1. They may undergo the activity of remodeling enzymes as occurs in plasma. Among those, LCAT, the cholesterol esterifying enzyme called LCAT, and the phospholipid transfer protein have been identified in the brain as well, in order to generate mature spherical particles that are finally able to deliver cholesterol to neurons through an internalization process mediated by specific ApoE recognizing receptors. The notion that cholesterol metabolism dysregulation is associated to Alzheimer's disease is not novel, but you can easily recognize in this slide here several factors actually associated to Alzheimer's disease are also genes encoding for proteins involved in brain cholesterol transport, such as apolipoproteins, lipid transporters, and ApoE receptors. So the hypothesis that we are making is that brain cholesterol transport may be defective in Alzheimer's disease, eventually leading to a loss of cholesterol internalization by neurons, finally ending to neurodegeneration. And actually, to better understand these potential defects, we dissected out this process in 3 main different steps. The first one, which is the HDL-mediated cholesterol efflux from astrocytes. The second one, which is the HDL remodeling and maturation in the cerebrospinal fluid. And the third one, which is the HDL cholesterol-mediated cholesterol internalization by neurons. Concerning the first step, it has been demonstrated that the apolipoprotein A4, which is the main risk factor for Alzheimer's disease, is actually less able to promote cholesterol efflux from astrocytes as compared to ApoE3, as you can see here by comparing these 2 first bars. And actually, the mechanism seems to be related to a trapping of the protein ABCA1 within the cells, so that ABCA1 is not anymore able to modulate, to promote cholesterol efflux from astrocytes. Again, concerning the first step, so the HDL-mediated cholesterol efflux capacity, we have demonstrated that particles, HDL-like particles present in the cerebrospinal fluid of Alzheimer's disease patients are defective in terms of promoting cholesterol efflux, specifically from the transporters ABCA1 and ABCG1. And interestingly, these cholesterol efflux pathways were significantly correlated to the usual neurobiomarker used for Alzheimer's disease diagnosis, such as the levels in the CSF of beta amyloid 142, which is the peptide that usually accumulates into brain parenchyma, as well as the levels of total tau and phosphorylated tau, which are an index of neurodegeneration. More recently, in collaboration with Professor Laura Calabresi from the University of Milano, we have also demonstrated that particles present in the cerebrospinal fluid of another cohort of Alzheimer's disease patients are defective in specifically promoting cholesterol efflux from human astrocytes. And we also demonstrated that this defect is even worsened when we pre-treated human astrocytes with beta amyloid fibrils that have been used in vitro in order to mimic an Alzheimer's disease-like condition. Actually, in this same cohort of patients, we also were able to detect specific defects in plasma HDL functions, and in particular, we saw that the HDL capacity to promote cholesterol efflux through the transporters ABCG1 and ABCA1 was significantly lower in Alzheimer's disease patients as compared to control subjects. Coming to the second step of the process, so the HDL remodeling and maturation in the CSF, again in collaboration with Professor Laura Calabresi, we demonstrated that the cholesterol esterification process is significantly hampered in Alzheimer's disease, clearly suggesting how there is an impairment in HDL maturation in cerebrospinal fluid, eventually leading to a lower supply of cholesterol to neurons. I will skip this slide, which is basically a confirmation of our previous study. Finally, coming to the last step of the process, mature HDL are finally able to deliver cholesterol to neurons through the activity of specific receptors, such as LDL receptor, but also others, including LRP1 or the ApoE receptor 2. And when we talk about LDL receptor, we cannot mention an important protein that has been widely studied at hepatic level, which is PCSK9. This protein on hepatocytes is able to degrade the LDL receptor, thus leading to an impaired clearance of LDL from the circulation and eventually ending to hypercholesterolemia. And based on this mechanism of action, several strategies have been developed, including the antibodies, very effective antibodies anti-PCSK9. Actually, PCSK9 seems to be able to degrade also the neuronal ApoE receptors responsible for cholesterol internalization, as you can see from the results of one work published last year, in which PCSK9 overexpression in neuronal cells is associated to a significant decrease in protein expression of the ApoE receptor 2 and the LDL receptor. And actually, PCSK9 is now object of intense investigation in order to find out a potential role in the brain, in the central nervous system, and in order to highlight a possible involvement in several diseases, including Alzheimer's disease. In this regard, we and others have demonstrated that this protein is increased in the brain of Alzheimer's disease patients, as you can see in the left panel, and also cerebrospinal fluid levels of this protein are increased in Alzheimer's disease patients, as you can see in the right panel. And again, in vitro, we have demonstrated that actually PCSK9 is able to impair the HDL-mediated cholesterol internalization into neurons as a possible consequence of the degrading action on ApoE receptors, ending to a net reduction of intracellular cholesterol content with potential deleterious consequences. And again, another toxic effect of PCSK9 was the capacity to worsen the neurotoxicity induced in neuronal cells by the incubation with beta-amyloid fibrils, again used to mimic in vitro an Alzheimer's disease-like condition. So to summarize the first part of this talk, we have seen how we have highlighted defects in brain HDL capacity to promote cholesterol efflux from astrocytes. We have highlighted defects in the HDL remodeling in the cerebrospinal fluid, and also we can state that HDL-mediated brain cholesterol internalization may be impaired in Alzheimer's disease due to increased level of PCSK9, ending up in less flux of cholesterol to neurons and possibly leading to neurodegeneration. So the other question is that can brain HDL may be considered a pharmacological target? And in order to answer to this question, I will go through the available literature data that are suggesting how at least 2 steps of this process may be enhanced by pharmacological strategies. Concerning the first step, several HDL mimetic peptides have been developed, direct ApoE or ApoE1 mimetic peptides that possibly acting through an enhancement of brain cholesterol transport may also have favorable effects on other features of Alzheimer's disease, such as neuroinflammation or the deposition of beta amyloid into the plaques. And in fact, one of the most promising peptide, the CN105, was able to improve beta amyloid pathology and to ameliorate the cognitive functions as evaluated by specific behavioral tests in Alzheimer's disease mice model. In a similar manner, the infusion of human recombinant ApoE1 Milano, you have heard from Anna about ApoE1 Milano, it's a natural variant of the ApoE1 discovered by our colleagues at the University of Milano with several cardioprotective properties. Actually, this apolipoprotein in this work was also able to ameliorate memory performances in mice, in Alzheimer's disease mice model, and again to ameliorate, to reduce a beta plaque deposition in these mice. And interestingly, these authors hypothesized not a central effect because they don't see any APOE1 in the brain after infusion, but they detected an improvement at the blood-brain barrier, since the infusion increases the expression of this protein called occludin, which is a tight junction protein responsible for blood-brain barrier integrity, and reduces the ratio between these two factors, which is a well-accepted marker of endothelial damage, so suggesting an additional protective effect, not at the central level, but at the blood-brain barrier. Probucol is another very interesting molecule. It's an hypolipidemic drug, which was able to increase, as you can see in the left panel, the levels of ApoE in the CSF, and in parallel was shown to ameliorate cognitive functions in humans, in a small cohort of patients with mild to moderate Alzheimer's disease. And now Probucol is under phase 2 trial investigation, trying to prove its effectiveness as an Alzheimer's disease counteracting drug. Finally, moving to the last step of the brain cholesterol transport, a possible strategy to enhance the cholesterol delivery to neurons may be the inhibition of PCSK9. And in this regard, we have shown that the genetic absence, the genetic deletion of PCSK9 in a severe Alzheimer's disease mice model was able to ameliorate the cognitive performances after repeated training, as you can see in the panel A, and this would occur through a reduction of the beta amyloid plaque deposition, as you can see in the panel B, and through an attenuation of neuroinflammation, as you can see in the panel C. So, to summarize also the second part of the study, we have seen how HDL-based therapies may improve Alzheimer's disease as evaluated in both animal models and in human. And I must mention that there are very interesting data on Cuban polycosanol on the effect on Alzheimer's disease actually in mice model of Alzheimer's disease as well. And also that the inhibition of PCSK9, possibly with molecules acting directly to the brain, may be a valuable strategy to improve Alzheimer's disease, although it has been so far demonstrated in animal models. So what could be the conclusion of this talk? And the take-home message is that we have seen how brain HDL-mediated cholesterol transport may be a relevant aspect of cerebral lipid metabolism, of course, not the only one. The multiple steps of this process may be defective in Alzheimer's disease. We were able to detect some defect also in the plasma HDL of Alzheimer's disease patients in terms of function and notably in cholesterol efflux capacity. And all the data together may suggest that targeting HDL and HDL-mediated brain cholesterol transport may be a promising pharmacological strategy for the future development of valuable therapeutical opportunities. Finally, let me thank all the people that contributed to this work from the University of Milano, Parma, Modena, Reggio Emilia, and Padova. Of course, the research support, and I would like to give you a greeting from the University of Parma and from the research group where I'm working on, headed by Professor Bernini. Thank you for your kind attention.